my general interest is trying to understand what drives the patterns and processes of biodiversity. And so I'm showing you this picture because it reflects what we find in the tropics. So when we go out of this room, we see thousands of species and colors and shapes, and those are the end result of adaptive diversification. In fact, Darwin referred to this as the entangled bank. And so my main research interest, my research questions are trying to understand what drives these processes and what promote that entangled bank that Darwin referred to. And so, and generally, I'm interested in understanding what drives diversification, when the species are formed, what keep those species coexisting in one area, and what are the processes that limit those diversification. And to me, these are probably one of the most exciting ideas about how we can understand diversity, especially in the neotropics. And so one conceptual framework that he used to study biodiversity is what we call the adaptive landscape. And this is a very simple model where we can try to see how the mean fitness of the population varies as a function of the combination with traits of those populations. In other words, how the reproductive success of the species will be the result of variation in morphological or phenotypic characters of those populations. And so an adaptive landscape is formed by what we call adaptive peaks, and each one of these adaptive peaks reflects areas of high reproductive success in nature. And so those adaptive peaks are separated by what we call thickness of low valleys. And so adaptive diverging stars, when single species or members of a single species manage to colonize different adaptive peaks in adaptive landscapes. And so the way we see is that nature is composed by thousands of different adaptive peaks, and those are colonized by different species. And those species go there by adapting to those peaks. And so this is a really cool framework to understanding diversification, because it actually allows us to make predictions that we can go into nature and test them. And so being in the tropics, it is a remarkable way it's a marvelous opportunity to go and then test for this framework. And so the first system, or one of the systems I use, or model systems I use to try to understand diversification in nature, is what we call Darwin's finches from the Galapagos. And so just to remind you the story, most of you probably are familiar with this. Somewhere 2 million years ago, a group of birds from main island Ecuador managed to colonize the Galapagos Islands. And so this is a distance of around 1,000 kilometers. And so within 2 million, we now have 14 new species of what we call Darwin's finches. And some of the most interesting features of this, what we call adaptive radiation, is that from that single ancestral species, we now have a whole array of new descending species, and every species has a different big size and shape, which is adaptive to exploit different facets of the environment. And so this model represents a really interesting way for us to understand what drives diversification in nature. And so one of the problems that we have when we try to ask this question is that when we go into nature, we only see the end result of the process of diversification. So we see the species around us, but we're not able to see the process that drives divergence in action. And so I was lucky that when I went to the Galapagos, we managed to basically see this process in action, and I'm going to tell you that story and try to convince you that we can actually go and see evolutionary process in action, and we can learn about those. So Charles Darwin, in 1839, he actually described this process of adaptive diversification. And he wrote that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends. It's a very poetic way in how Darwin described. And I'm using a picture of young Darwin because that actually happened. He wrote this phrase in the second edition of the Voyage of the Beagle. So was right after they finished the travel around the world, basically. And so This is the first written record of Darwin talking about evolutionary processes. And basically, what he meant by that is that, and here showing a picture of what we call Darwin ground finches. So it's just a little part of that adaptive radiation that I showed before. And these are granivorous finches. They feed on different seeds on the ground, mostly. And so you can see a really nice gradient in big sizes and shape. And again, every species has been modified for different ends. We have the small ground finch that eats small and soft seeds, the medium ground finch that eats medium-sized seeds, and we have the large ground finch that feeds on those really large and hard seeds. And down here we have what we call the cactus finch that has a really interesting looking bill that uses to exploit nectar and flowers of cactus plants. And so we can really see very precisely what Darwin meant that by when he said that all these species have been taken and modified for different ends. Even more interesting, and show you the big size distribution of those granivorous finch. I'm not putting here the cactus finch that I showed before. I'm just showing you the ground finches, the ones that eat granivorous, the ones that eat seeds in the ground. And so there is really wide distribution in big sizes and shape. And I can map each species onto that distribution. So we have the small ground finch, the medium ground finch, and the large ground finch. We can see very nice separation between the small and the medium. The large is really less common in the island. But something really striking here is that within the medium ground finch, there's this really wide distribution of big sizes and shape. In fact, when we do the math on this distribution, we realize that this wide distribution within the medium ground finch actually fit 2 modes instead of 1. So this distribution is actually bimodal. And let me illustrate that with some pictures. So this is a small ground finch within the medium ground finch. This is a small morph, there's 2 morphologies within the medium ground finch. And at each extreme, we have the small and the large ground finch. And so this is really striking to us because when we were in the field, we were trying to identify those birds into 3 categories, small, medium, and large. However, we realized that we were getting these birds on the net, and these birds were kind of small, but it's a kind of medium, intermediate bird, but also small and large. And so very striking to us. And these are some of examples of the seeds they normally eat in the field. And so I started wondering, maybe we're actually observing the initial stages of adaptive divergence within these populations. And if so, that might represent one marvelous opportunity to test what are the processes that drive this divergence in nature. And so this work was done in the Galapagos. So this is Panama down here, very close to the Galapagos. It's the Galapagos. And the work was conducted on Santa Cruz Island, which is the 2nd largest island of the archipelago. And also, ecologically, it's one of the most heterogeneous islands of the archipelago. And so this is just a picture so you can get an idea about how the vegetation varies within the same island. And so presumably, that variation in environment could lead to selection that will promote divergence in natural populations. I'm gonna try to test that for you. And so how do we do this work? So my research in general combine theory from ecology and evolution. We use population genetics or molecular tools. And we do also a lot of field work. In fact, here I'm in the field setting up nets. We collect thousands of birds in different places in the island. For each bird, we take a lot of measurements, big sizes and shape. We ask each bird to bite on this little device very gently, and that device allows us to estimate the bite force. And that's a very interesting trait because that's associated to the size of the seeds they can crack. And so we can do analysis on those. We collect blood samples to do genetic analysis, and every bird we band with a combination of color rings and color bands. And Darwin's finches are in general very tame, so we can follow them in the field and see what they do. So see what type of food they eat, they feed on, and see who they mate with whom, for example. So we can do all these sort of studies. And we can also estimate survival. So across the years, we can see which birds survive and who dies after the end of each sampling period. And with those data, we can estimate both the force of natural selection in nature and the direction of selection. And this is very few opportunities. And I also do a lot of field work, collecting seeds. Basically here, I spend months in the island, literally crawling on the ground, counting seeds, just to have an idea of what the distribution of resources are on the island. And so, based on the conceptual framework that I introduced at the beginning, we can have several predictions about what are the processes that drive divergence in nature. First, we start with variation in the ecology, and that will promote ecological divergence, which can presumably lead to what we call disruptive selection. It's selection pushing populations into different directions. We can also get data from story mating, basically what are the mating pattern of the population. And we can estimate whether we have any signal for reproductive isolation and basically or essentially be observing the rise of new species on the field. So I'm going to show you some of the data that we collected. This is just diet data. So we observed thousands of birds and we're looking at what they were feeding on in the island. And so this is the axis of diet. The black dots represent the diet for that large morph that I showed before. And the white dots represent the diet for the small morph. And interestingly, there is a very nice separation between the two morphs in the type of food that they eat in the ground, in the island. And this is, when we do the study, this is statistically significantly. So that means that we have some evidence for ecological divergence between the small and the large big morph within the medium ground finch. Now, one of the questions that I got afterwards, okay, we have divergence in ecology. Is that enough to promote selection or disruptive selection in natural populations? And so we went back to the field, we went back to the Galapagos, and we estimated survival across many years. And so I'm showing you data from survival for 2 years as a function of big sizes, of big morphology. All these blue spots down here show the initial population, the one that we sampled at the beginning of our study. And here are all those birds that survived at the end of the study. So with this data, we can we can feed a statistical model of something that we call a cubic spline and see where is the probability of survival of the two big morphologies, basically. Something really striking here is that it seems that either the small and the large big morphologies within that population are doing better, are surviving better. So somehow, selection is going against the intermediate-sized birds. Those are somehow doing really doing bad in the population. So with this data, we show that, in fact, those two modes, again, correspond to the modes of the small and the large big morphologies within the medium ground finch. And in fact, this data was one of the first estimates of disruptive selection in nature that we know of. So the way that nature is starting to promote divergence in natural populations. Now, something that is interesting is that these birds are coexistent in the same area. So presumably they can mate with each other, right? This all random mating. And if so, that divergence that we're observing might not be sustainable in time. And so we went to the Galapagos again with my colleague Sarah Huber, and we followed birds. So we're and try to estimate how many pairs we can get in the field, and who was mating with whom, and what are the pattern of mating in this population. And so here are the female, which is a little bit brownish. This is the black male. And the data that I'm showing you here are all the pairs that we observe within one field season. And this is how those pairs vary or are formed as a function of big morphology. So interestingly, females with small big sizes mate almost exclusively with males with small big sizes. On the other hand, the females that have larger big sizes would prefer to mate with males that has larger big sizes. And so there is what we call assortative mating within each population. And so to us, this is another piece of evidence suggests that not only ecology, but natural selection and sexual selection might be promoting divergence within these populations. And so we have evidence for that, and now we want to try to test, is there evidence for reproductive isolation, which is the kind of the final step before we get new species. One way that we can test that is by estimating how much gene flow, how the genes move between the small and the large big size morphs. And so I did that by using molecular markers, microsatellite markers, and this is just the result of the distribution, a factorial analysis of the distribution of those genotypes within those populations. And so something really clear from this data is that we can see two clear clouds of points, yellow and blue, yellow corresponding to small morph and blue corresponding to a large morph. So interestingly, there is still quite a bit of overlap. And so these are genetic overlap between the two types at the same site. And in fact, when we do the stat analysis on those genotypes, we realize that there is significant genetic differences between the types. But these FST values, which is, it gives us an idea of the genetic distance between the two morphologies, are fairly slow. And here you can see it's 0.02. And this is very exciting to me because that's what we expect to see when populations are at the initial stage of divergence. And if so, we probably are observing this process in action, and that gives us a really nice window to try to understand what drives diversification in nature. Now, so we have some evidence for partially reproductive isolation within the same site, within the same area. Now, I hope I have convinced you so far that studying species or population that are undergoing the initial stage of divergence is a powerful model to understand diversification and biodiversity in general. Of course, as happens in our field, we sometimes get more questions than answers. And for example, here again, these are the two big size morph, small and large, within the ground finches. You might be wondering, how does that bimodality, how is that adapted divergence fit in within the whole radiation of finches, right? Or maybe, and that's one of the questions I always get when I present this data, is okay, and I hope I don't spoil it for you, is that, okay, where those two big morphologies actually came from? There's two possibilities. So one possibility is that maybe they diverged in the different islands in the archipelago, and then came into secondary contact now on Santa Cruz Island, and so they have a previously divergent, and now they're competing on the same site. That's a possibility. Or maybe they actually arose on the same site. And so one way we can try to test these two hypotheses apart is by using next generation sequencing and genomic data to understand the patterns of gene flow across the species. And so in a collaboration with my colleague Jaime Chavez from Ecuador and from other collaborators from the US, we use RAG-tag sequencing, where we sequence randomly those three species, and we use RAG-tags, which are random sequence across the genome, basically. And we want to understand what are the patterns of variation, of genetic variation across the genome of those species. And so the two morph within the median, and the large one. And this data represents the probability of inclusion, basically the variation of genetic variation across the genome. Each color represents a different scaffold of the genome. And this data is an analysis of why genomic association analysis is as related to big morphology. So this dashed line here represents a line of neutral genetic variation. That suggests that everything below this line shows no variation, no differences across the three species that I showed before. And so something very striking is, okay, we can actually observe that these three bird species genomically are really similar across the genome. There's not much variation in it, except for few spots on those axes that show those regions of the genomes that actually explain most of the variation or the difference between the three species that I showed you before. And more interestingly, those regions of the genome are associated to big morphology, which is the main trait that we've been looking at. So this suggests that probably the adapted divergence within this population of this species is associated to a small island, what we call island of divergence in the whole genome. And those islands are regions of the genome that are under strong selection. And that selection is probably what ultimately drives to divergence. And so I'm going to show you just the variation in one of these genes, as an example, the DLK1 gene, which is associated to big morphology. So I'm going to show you the allele variation in that gene. So this is the allele variation within the DLK1 gene as a function of big morphology. So we have the small, the medium, and the large, small in orange, medium in green, and large in blue. Something very remarkable about this data is that almost all the small groundfinch are homozygotes for that T allele within that same locus, same gene. At the other extreme, all those large, big morphologist birds are homozygous for the other alternative allele, the C allele. And within the medium ground finch, we actually have a combination of both. Some of our homozygote, heterozygotes, but right in the middle of the distribution, those intermediate birds are the ones that have been selected against in the population, are generally heterozygotes. And so this is really, we start to understand what all this data means, but something that we learn from this is that the integration, we know already that the cellular gene flow, the whole genome is very similar. So the cellular gene flow across the three species. But the integration of the gene flow of allele variation associated to adapted genetic variation seems to be one of the causes of that large variation that we observed within the medium ground finch. And so basically, or essentially, we're identifying a new factor where the integration between different species can also drive diversification in a natural population, can drive the genetic variation or the adaptive genetic variation where selection will act on and will promote divergence. And so I hope I convince you that we can actually observe the process of adaptive divergence in action in natural population. Now I'm going to tell you a quick story of something that we're doing here in Panama now, where we keep working on birds. And these white-breasted wood wren are really common birds that are distributed across the Americas, basically from northern Argentina up to Mexico. And so there's quite a bit of variation, and the variation in these birds is not associated to big morphology, as in the case of finches. In this case, it's associated to color plumage, of coloration on these birds. And so this is the distribution of those birds. Each of those names represent subspecies or subpopulations that people have named as a subspecies within the same species. But really, we don't know what is going on within that whole range of the, in this case, the distribution of the species. And so we actually collected data. We will be able to get data from all these, or many of these populations across the Americas. And we also collected quite a bit of birds within Panama to understand, okay, how much variation is there, and what are the implications for divergence within these species. And so with this data, and with the help of my PhD student, Celestino Aguilar, that's his PhD thesis in INDICASAT, we're applying these new genetic markers that we call ultra-conserved elements. And we extract those, we get those by using sequence capture in the lab. And ultra-conserved elements are regions of the genome that are really conserved across species. And the neat thing is that they're very conserved, but as you move away from those regions, you start getting variation. And we can use that variation as genetic markers that we can apply then to ask biological questions in different populations. And so why we are, or Celestino is basically doing this work, where we have use next generation sequences to amplify those UCEs or ultra-conserved elements in these populations. And so I'm going to show you a really quick, basically straight from the oven data that we have been generating at INDICASAT with Celestino. So this is just a phylogenetic tree as a coalescent model that basically wants to understand what is the evolutionary evolutionary history across all these populations across the Americas. And so here we can see all the population. We have samples from Ecuador all the way to Panama and Mexico. Something that is very interesting is that all this dark, dark, like green defined spot represents very stable evolutionary relationship. That means that it doesn't matter how many times we run this analysis. We did this for 5 million times. We get the same topology, the same structure, or the same relationship across populations. However, we can see here some really like light lines here, and that represent species where there might have been recent mixing or integration of genes. And so that basically alters the need patterns that we had. And so within this tree, we have Panamanian populations that are over here and also over here. So somehow they don't cluster together. And so the next thing that we did, what Celestino actually did, is that, okay, we want to focus in those Panamanian populations and we're going to look at the patterns of integration of gene flow across populations within Panama. And so very remarkable this data is that we have in yellow all the Panamanian population from eastern Panama, and in blue the population from western Panama. And there's a really sharp point here where the two are mostly separated. But interestingly, when we do the structure analysis, and these lines represent the genetic makeup of each one of the individuals in the population, we realize that within this area right here between Cerro Azul and Ashote, like in the center of Panama, there's some integration coming in. In fact, we have genes coming from the yellow species into the blue species. And those genes actually move even a little bit far into western Panama in the area of Bocas del Toro. So again, with this data, we're suggesting that adaptive divergence or adaptive integration, or the integration of genetic alleles or genetic variation associated to adaptation might be one of the new ways that we can think of the evolutionary processes that drive divergence in natural population. So hybridization to many people is a process where that probably hampers biodiversity because we have independent species. And once those species come into secondary contact and they start to hybridize, they will collapse into a single species. What we are showing with this data is that that process of integration between even separated species could promote further divergence. And so we would like to keep expanding this data into other systems in Panama to basically start to understand what drives this divergence in neurotropical environments. And so this is Celestino, and all the data I'm showing you lately belongs to him, basically. And we're still now trying to write these results. Finally, I would like to thank you, all my collaborators that helped me a lot during this process, and all the funding institutions that provide the money to do the work. And thanks for your attention, and I'll be happy to take any questions, if any.
Pregunta.
Thank you very much for such an extraordinary conference. I mean, I'm fascinated. I'm an evolutionary-oriented person, definitely, so I learn a lot from that. It's much more than a question, a thought. I would like to hear what you think about. Hearing you and diversification and everything that you have set up, I think about mosquitoes. And the first thing that came up to my mind was the populations and the natural adaptation that the mosquitoes might have. And we know that in Panama we have various serotypes of mosquitoes. And we know that recently it was on the liberation of genetically modified mosquitoes and all those type of things. So my question to you is, what do you think about this type of genetically modified mosquitoes being thrown in the environment vis-a-vis the natural process that takes place on evolution and adaptation of mosquitoes and the prototypes or serotypes, using the right words from a scientific point of view?
Yeah,
Okay, thank you.
Yeah, so she was wondering about mosquitoes. Mosquitoes are also a really cool system where you can ask questions about adaptation and diversion. Like, they evolve really, really fast. And that's a nice system that you can actually study using the same framework I have shown you. With regard to the release of genetically modified mosquitoes, I think one of the tricks is that, or essentially, as we understand it, those mosquitoes are not able to persist in the populations. And if so, that case, it wouldn't alter the genetic makeup of the population. But if we assume that that could happen, that there's a possibility that those mosquitoes will be able to hybridize with the normal wild mosquitoes, we might be able to be introgressing new adaptive genetic variation that was like new in the or is new in the population, and that probably will produce a new evolutionary trajectory basically in the mosquitoes. That could be a really interesting possibility. But as far as I understand, those mosquitoes won't persist in the population, at least those genetic new strains in this case. But yeah, that's a very interesting possibility.
Thank you very much.
Yeah, you're welcome.
Congratulations, Luis. It was a great presentation on work. My question is simple. What exactly is a cladogram? Is that a new tree, or a new type of tree, or?
Yeah, a cladogram it's kind of cloudgram. It's a new type of tree where you you repeat the same tree or build the tree like 5 million times, for example, and you cluster them all together. And so that's why you see all this pattern of, you see the clouds of tree around the planet, the whole main framework.
You put all the trees together?
Yes, basically, yeah, that's how it works.
And you use a scale of transparency or different color intensity for the branches that are not supported?
Yes, yeah, you can support. In this case, what we want to know is how stable is the main topology of the tree.
Okay.
If you build it like 5 million times. And so it gives you a neat idea of the main branches that are really stable. Doesn't matter how many times you do it. And those branches that kind of fall off that pattern are indicating of recent, in this case, population intermixing, basically.
Okay.
And so that's where we're looking at.
Is there a software, a specialized software to build them? Or?
Yes, it's called MADP, map tree. And yeah, we can send it to you.
Okay,
You can play with it. Yeah.
Yeah.
This
Thank you.
software do it, yeah.
Una pregunta más.
Hi, very interesting talk. So I was thinking about where you showed the medium-sized beak family, and it's separated into small, mid, and large. So you showed in your graph that the mid-range has the lowest probability of survival. I was wondering why, because I would think that the ones in the medium group have a medium-sized beak, so they could basically compete in two groups. They could compete for all of the small seeds, but they could also compete, because I saw the seeds that you showed had quite a range, so they could compete with a large-sized beak for the smaller ones. So essentially, instead of the small and the large, which are competing within their own niche, the medium-sized ones could possibly compete in both. So you might expect the opposite, that it would be more fit. So what is your current theory to explain this, for why?
So something that I've done quite a bit is estimating the range of distribution of seed sizes and shape. So yeah, we do have a really wide range of seeds types, but somehow we have less intermediate sized seeds. And so the idea is that those intermediate birds, they fall in that kind of gap where there is not much seeds, and the ones at each extreme are really efficient at exploring those seeds. And so basically competition pushes those apart. And just by the distribution of seed types, it's better to have either small and large big size. but again, not too small or not too large. And so because that creates a trade-off between the size of the bill you have and the type of seeds you can break. And so yeah, that's one of the reasons that we know is affecting the survival of intermediate-sized birds. Thank you.